All right, welcome to Let's Get to the Marks. Today's question is a biology A-level walkthrough, and it's about describe the process of glycolysis. Some people call it glycolysis. It's worth 4 marks. Let's get into it. Now, in order to answer this question properly, I'm going to explain the process of glycolysis. I recommend you pause the video, have a go at writing down your answer on a scrap bit of paper, and then unpause and watch the walkthrough. Let's go. Okay, so glycolysis starts with a glucose molecule in the cytoplasm. So not inside the mitochondria. Now, glucose, if you're a biologist, you have to remember it's a 6 carbon molecule. And what happens to the glucose during glycolysis? Well, one ATP molecule, adenosine triphosphate, is turned into adenosine diphosphate. It loses an inorganic phosphate represented by the PI, and that phosphate is joined to the glucose. What do you think you call glucose when you add a phosphate to it? You just call it glucose phosphate. Dead easy peasy. So one ATP molecule was used to add a phosphate to the glucose. Guess what happens next? A second ATP molecule is broken down into adenosine diphosphate, or ADP, and inorganic phosphate, PI, we write that as. You can just write P. And that inorganic phosphate is also added joint to the glucose phosphate. So now the glucose molecule is exactly the same as it was before, but it has two phosphate atoms, or phosphate groups, sorry, they're not atoms, they're groups, added to them. Okay, now once you add two phosphates to a glucose, it's now called hexose bisphosphate, or bisphosphate, can't really say bisphosphate, but bi means two. So it's basically saying hexose means six, a 6 carbon sugar with two phosphates added. So we now call the glucose hexose bisphosphate because two phosphates have been added. Now, this is an unstable molecule, and this breaks down on its own accord. It doesn't need anything added. It is unstable, so the bonds in it will break, and it will form a more stable formation, and that is two molecules of TP. And TP is known as triose phosphate. So each of these two molecules has 3 carbons inside it. Now, what we call this whole first bit, I'm going to outline this first stage of glycolysis. Okay, so this is the first stage of glycolysis. Obviously, I'm explaining this so that you can answer this question to describe the process. And this first stage of glycolysis is known as the phosphorylation of glucose. And phosphorylation is a really important role of ATP. So you might get a question in A-level biology asking you, what can ATP be used for, or why is ATP important? One of the important things about ATP is it can be used to phosphorylate molecules, which means to take one of its phosphates off when it forms ADP and give that phosphate to another molecule to phosphorylate it. So my first answer here in describe the process of glycolysis should be, well, the first thing that happens in glycolysis is glucose is phosphorylated. Or I could say the phosphorylation of glucose. Okay, now you could describe that and you'll still get the marks. You could say, oh, glucose has ATP breaks down to ADP plus PI, and the PI joins the glucose and it becomes glucose phosphate. That's all long. You can just sum that up by saying glucose is phosphorylated. Now let's get on to the second section. What happens once it's become TP? Well, here for ADP stands for adenosine diphosphate, join with 4 inorganic phosphate groups represented by the symbols Pi, and they become 4 ATP. So think about this. Glycolysis used up 2 ATP in the phosphorylation of glucose, but then when we get to the TP, what we're going to do to the TP is oxidize it. We call this the oxidation of triose phosphate. And during the oxidation phase of glycolysis, the oxidation of triose phosphate, we make 4 ATP. Now, when triose phosphate is oxidized, 2 hydrogen ions are stripped off of the TP molecules and they're added to NAD. Now, NAD, you don't need to know its name. You can just use capitals, the abbreviation for this, but it's nicotine adenine dinucleotide. Whew, you won't have to say that, trust me. But these 2 hydrogens are added to NAD and they make 2 reduced NAD. Now, it's fashionable nowadays to call NADH, reduced NAD, but essentially you've just added a hydrogen ion to NAD, so you could call it NADH. Back in my day, we used to call it NADH, and it made a lot more sense. NAD is reduced by adding hydrogen to make NADH. Now we call them reduced NAD. Also, during this process, the TP becomes two pyruvate molecules. They still have 3 carbons. All that they've had is hydrogen ions stripped off of them to reduce the NAD. So what do we call this second stage? That's the whole of glycolysis right there on the page. What do we call this second stage? What has happened to the TP? Well, the TP has been oxidized. So we call this second stage the oxidation of triose phosphate. And 3 things happen during the oxidation of triose phosphate. You get 2 molecules of reduced NAD formed, you get 2 3-carbon pyruvates formed, and you get 4 ATP formed. So my second mark in my described glycolysis should be TP is oxidized, or TP undergoes oxidation, or the oxidation of TP, and that will get you a second mark. And then all you need to do is explain what's formed in the oxidation of TP, because that's the main bit where all the cool stuff gets formed that's needed for all the other reactions in respiration, anaerobic and aerobic. So we get 2 pyruvate molecules form. That's going to get you a mark. Now those pyruvate molecules can either go to aerobic respiration in the link reaction, or they can go to anaerobic respiration where they help reform NAD to allow glycolysis to continue. But we also get 2 net ATP form. Remember, we used 2 ATP and we made 4 ATP, so we call that a net production of 2 ATP. Some organisms can survive alone by glycolysis. They don't need to do the link reaction, the Krebs cycle. Anaerobic microorganisms just use glycolysis and aerobic respiration to reform NAD. So they can survive off just a little amount of ATP. Obviously, they're not active like animals and plants, but there we go. Right, 2 reduced NAD are also formed, and these reduced NAD are really important because they're coenzymes, they're hydrogen carriers and electron carriers, and they later on are used in oxidative phosphorylation in aerobic respiration. And they're used in anaerobic respiration, these reduced NAD, to reform the original NAD so that glycolysis can continue. Now, this would get you 4 marks. I know I've put 5 ticks there. And what I'm trying to highlight here is when you're revising glycolysis, you might find this too much with all the huge biology A-level content. So what I'm saying is, rather than memorizing this whole process, you'll probably score all of your marks on questions to do with glycolysis by just remembering that glucose is phosphorylated. Where do the phosphates come from? They come from ATP, but also that TP is oxidized. So they're the 2 main things that happen in glycolysis: phosphorylation of glucose, oxidation of TP. Now, what is made in glycolysis? 2 reduced NADH, 2 pyruvate, and 2 ATP. They're the 3 products at the end of glycolysis. Okay, so if you're asked about glycolysis, personally, I would just memorize, yeah, glucose gets phosphorylated, TP gets oxidized, 2 reduced NAD is made, 2 pyruvate are made, and 2 ATP are made. Now, it is good if you understand what all those products are used for, and you can go into the depth and learn all the details, but I would definitely, my starting point would be to memorize those 5 key points. All right, guys, I hope that helped. Don't forget to share, because that's nice, and hit the like. A lot of people don't bother hitting the like, but it really helps my videos out if you do. Punch up the bell, subscribe. Let's get to the marks. I'll see you guys in the next video.
You're still here? It's over.